first you have to identify the particular locus you want to modify, and then with the help of endonucleases enzyme. Earlier I mentioned that engineered endonuclease, because they are not natural. Something has been added in that so that they can meet our criteria. So there is a protein which is going to identify the particular locus that we want to add it, and with the help of these endonucleases they are going to cut. So what are the different uses of gene editing? Gene knockout. You can completely eliminate the particular gene you don't want. Say for example, a particular gene which is causing a abrupt phenotype in the plant or in any animal or something. So for example, it is causing a particular disease. So with the help of genome editing, you can knock out that particular gene so that you are not going to get the same that phenotype in the next generation. Gene knock-in. You can insert a particular gene. You can next is gene tagging. You can tag, means what, where in the genome which this particular gene lying in the, where it is lying, what is the position of this gene, and what is the function of the this gene. Okay, so with this is the gene tagging. You can tag a particular gene in the whole genome, and we can study the insertion and deletion, and also the we can identify the promoters. So if we'll see the different mechanism of the tools for the genome editing, the first one is meganucleases, next is zinc finger nucleases, and next is Talon, and last is the CRISPR-Cas9. So the first one is meganucleases. This was the first tool, or the we can say the starting of the genome editing has been done from the meganucleases. As I've said earlier, in genome editing there are two things important, and two things which are playing the important role. One is the protein part, which is identifying the genome portion where we have to edit it, and the second one is the nucleases which are going to cut that. So this is the first tool which is used for the double-stranded break, and it is mainly occurred in yeast and in the Chlamydomonas. What happened in this meganucleases, the enzyme binding site and the restriction site occur in the same unit. So basically there are two different units, one which is going to identify by binding to that particular site, and next one is the restriction site which is going to cut it, the particular site that we want to edit. But here in this, the binding as well as the restriction site occur in the same unit. So what are the different limitation with this? This is difficult to manipulate the DNA binding site. What is our purpose? Our purpose is just to identify and cleave. But when binding site as well as the restriction site present in the same unit, it is difficult to identify the targets and modify. That's why it is this is the limitation. And small recognition site, it could only identify the very small size of the DNA or the gene in the genome. That's why this is the limitation, and this has been used by the Gao and his co-worker in the maize for herbicide resistance, and this has also been used in the cotton for herbicide resistance and the insect resistance. The next mechanism or the next technique in this is zinc finger nucleases. So here you can see in the name itself, it is indicating that there is a zinc finger. A zinc finger are nothing but these are the protein which are going to identify the site which we are going to edit, and the nucleases are there. So these nucleases are going to cut the your gene of interest. So basically these are the engineered protein, this zinc finger, these are the enzymes. Okay, and nucleases are there which is fused with the zinc finger, and this protein has been made. So it consists of the two part, right? This is the zinc finger binding protein. So you can see here there is a zinc element present here, with the different finger-like structure. So these finger-like structure, where the zinc is an important component here, it is going to bind and characterize the site of the genome or the nucleotide of the genome which we are going to edit. These are the first identified as a DNA binding motif in African cloud frog. So this were first identified in this frog, and these are the small protein structural motif, and that is a coordination of one or more zinc ion that in order to stabilize the fold. So what is the function of zinc here? Basically, zinc stabilize the fold. That's why zinc is present here, and zinc finger motif has been identified or used for the genome editing. So and the endonucleases which is present here is the fork one. From the exam point of view, this is important that in zinc finger nucleases, the endonucleases used is fork one endonucleases, and it is naturally found in Flavobacterium oikinocoids. Okay, so this could be asked in the examination. So what happened here actually? This zinc finger motif is fused with the fork one endonucleases, and the system has been created. Okay, so here it is the same thing. So how this system is going to work? So here we can see these are the zinc finger protein motif, and this is the HOOK1 endonucleases. Okay, so first, what these zinc finger motif is going to do, based on the we can engineer these zinc finger motif. Okay, based on our requirements. So, for example, we have to target the gene for the flower color. So we first the requirement for the genome editing is that we should know the location and the sequence of the gene we want to edit. So based on that, we will modify our zinc fingers motif. So this zinc finger motif is going to bind in the particular gene of interest, and then it will restrict the DNA. Okay, these endonuclease, fork1 endonuclease, these will cut the double-stranded DNA, and now these double-stranded DNA, they will be handled by the two different mechanisms as I've discussed earlier, homologous recombination and the non-homologous end joining that I will discuss later on. So this is the zinc finger nucleus, where mainly the zinc finger motif along with HOOK1 endonucleases are going to edit the gene of interest. So what are the limitation of this is off-target effect. So what is off-target? So what happens, this genome is very large, okay, and we want to modify or edit a particular location in the genome, but it does not happen practically that only the gene of interest or the location we want to edit is only modified. This also goes and binds somewhere else in the genome. That's why it is also editing the different location in the genome that we really don't want, because it will unnecessarily bring some other changes. That's why this is called as an off-target effect, and this is because of lack of specificity, and we have to reduce it. The construction is cumbersome and the time consuming. The construction of zinc finger motif, and it's difficult in this case, zinc finger motif. Where it has been used, it has been used in the maize, soybean, and tomato for the different traits like herbicide tolerance, for physiological trait manipulation, and this particular disease. So the next mechanism or the next tool under the genome editing is the Talon. Why it is called as the Talon? Because it is TAL effector protein and endonucleases. Okay, so this is the effector. So what are the effector? Effectors are nothing but these are the different protein which are used by the pathogen for breaking the primary defense system of the plant. Okay, so these are the effector. So as I've told you earlier, there are two different units. One unit which is going to identify the gene of interest, and next unit, another unit is which is going to cut. So here in this case, the unit which is going to identify is the TEL effector protein. Okay, and then nucleases is maybe the same FOC1 endonucleases. Okay, so they are made by fusing the TEL effector binding domain, as I've discussed earlier, and which is going to identify this the gene of interest. And see, basically this TEL transcription activator like effector, these are the proteins secreted by the xanthomonas bacteria by their secretion system, and they help into activate the expression of the plant gene that aid in the bacterial infection. So this is the structure, molecular structure of effector. Uh, there is complete structure stretch of 34 amino acids, where the 12 and the 13 amino acids is RVD, repeat variable diresidues, means these residues are responsible for the DNA nucleotide recognition. So from the exam point of view, it can also ask that RVDs are present in which system. So RVDs are present in the Talon system of the genome editing. These are the two specific amino acid residue which is responsible for the recognition of the DNA, that is which we are going to edit. So this is nothing but these are the different bioinformatics tools through which we can predict the binding site and design the Talon protein. So this is the mode of action of the Talon. So this is the Talon protein, which is DNA binding domain, and this again you can see the endonuclease present here is the fork one. So in case of ZFN, zinc finger motif, and the zinc finger nuclease and the Talon, the endonuclease is same, that is fork one. So here we can see with the help of these TEL effector protein, we are going to identify the gene of interest. It will go and bind to the gene of interest we want to edit or modify, and this fork one will cut that gene of interest there. So here you can see this fork one has cleaved that particular thing, and there is creation of double stranded break. So these double stranded break, they will either they will be creating indel. Indel means insertion or deletion, and or if there could be a gap sealing by non-homologous end joining. So there are different ways through which this Talon method of genome editing could be used for the disease-resistant plant. So transcription activation of different R gene, we can it can bring about there are different R gene. R gene means resistant genes present in the plant. So it could help in the activation of transcription of these genes. Then repression of different susceptibility genes. Susceptibility genes are nothing but those genes which are responsible for the susceptibility to a particular disease of the plant. So with the help of Talon, we can repress or we can knock out those genes with the help of Talon. So mutation of the promoter of susceptibility genes. So we can bring about the mutation in the promoter of susceptibility genes, so that we are going to get the disease resistant plant. Next is the gene replacement. We can replace the gene with the help of this homologous recombination. So these are the different examples of using the Talon in the plant for the different traits. So here we can see in different plant species, these different gene has been modified, and this is the reference. These different group of worker have modify these particular gene in the different crop for the different uh, trait modification. Now the last, I cannot say the last system, the next evolutionary system of and the very famous and the very revolutionary system of the genome editing is the CRISPR-Cas system. There are lot of people and lot of research, not only in the plant system, also in the animal system. The CRISPR-Cas has been utilized a lot. So basically, the CRISPR-Cas system, the CRISPR has it has a full form of clustered, regularly interspaced short palindromic repeats. And why they are called as a CRISPR, I will tell you. So basically, this CRISPR is nothing but it is a bacterial immune system, and how it is working, I will tell you. First of all, let's see the history of the CRISPR. The researchers find CRISPR sequences in E. coli, but they did not characterize their function. So first of all, these CRISPR sequences has been first identified in E. coli. This could be important from the exam point of view. The CRISPR sequences are found to be common in other microbes. Other than E. coli, they are also present in the other microbe. Okay, coined CRISPR name and defined signature Cas gene. Along with the CRISPR, there are different Cas gene has been identified, and these Cas genes are responsible for the formation of Cas protein, which is endonucleases here. Okay, and then the first experimental evidence for the CRISPR-based adaptive immunity is given, and then in 2013, the Cas genome engineering in eukaryotic cells has been demonstrated. So this is basically the CRISPR-Cas system. You know what used to happen earlier. You know that there are bacteriophages, right? Those who are the viruses which is attacking on the bacteria. So what happens when the virus attacks on the bacteria? They deliver their DNA into the bacterial system of the plasmid, and when these their DNA or RNA has been attached into the plasmid, what happens? These are called as a now spacer molecule. Okay, the genetic material of the virus which is integrated into the bacterial DNA. Now it is called as a spacer. And what happens? This virus is very clever. So with the help of bacterial system, what it will bring, it will transcribe the genetic material of the virus, and again there will be the formation of different progenies of the virus, and different this virus will kill the bacteria and it will release. So what happened as a process of defensive system, this bacteria has developed some defensive mechanism. What it has done, so when these spacer molecule has been integrated into the DNA of the bacteria, when it is transcribed, the memory kind of memory has been created into the bacterial system. That say for example, first time when a virus has attacked the particular bacteria, but after subsequent attack of 3 to 4 times, what happens? These the genomic part of the virus, it will integrate into the DNA of the bacteria, and when virus again attack to the bacteria, what happens? The portion which was there in the genome of the bacteria, it transcribed, and based on the homologous recombination, because this the part which is already present in the bacteria, it will show the homology with the virus. So when it will, based on the recombination, homologous recombination, based on the homology, this will bind with the genome of the virus, and with the help of Cas protein, which was I was talking about earlier, with the help of this Cas protein, this will cut the particular virus genome, so that the virus is not going to proliferate further. It is not going to produce its progeny. So this is how the bacteria has adopted this CRISPR-Cas based immunity system. So this is in general when the this virus, it integrates its genomic DNA into the bacterial DNA. So what happens? It is going to produce the different progenies of it. But when the next time bacteria this virus attacks, so with the help of after this transcription, there will be the formation of the CRISPR-Cas complex. This is single guide RNA, and with the help of Cas protein, it will form a complex, and with the help of Cas protein, it is going to degrade the viral DNA. So what is most important thing here is the PAM. you can also ask the question like when same DNA is present into the bacterial system, then why it is not harming the bacterial system also? So it is not because the prime requirement for this cleavage is the PAM sequence. So PAM sequence is nothing but the proto spacer adjacent motif. Along with that particular DNA of the interest which is showing homology, adjacent to that there are three nucleotide sequences NGG, which is generally important for the CRISPR-Cas9 system. So if that sequence is present in the virus, that only it is going to, then only the Cas9 protein is going to cut, and it is only present in the virus, not in the bacterial system. That's why even though there is a homology showing between the bacterial system and the viral system, the bacterial sequences and the Cas9 is not going to cut the bacterial DNA sequence. Okay, so this is important here. So this is the CRISPR-Cas9 system. Here you can see this is the Cas9 enzyme and this is sgRNA, single guide RNA. So this single guide RNA, it is going to bind into the gene of interest, and you can see here the palm sequences. So this is responsible for the identification in the cleavage by the Cas9 enzyme. Okay, so after cleavage it will create the double stranded break, and after the double stranded break there are two different pathway we were discussing, that is NHEJ, non-homologous end joining, or HDR, homology dependent recombination. So what actually happens when there are double stranded DNA, when the two see these are the non-homologous ends now, so when they are joined, so when they are joining, so there are possibility that there will be creation of either indels, either insertion or deletion. Means when these ends are joining, there are possibilities that there could be some other nucleotide in between of them, or there could be deletion of some portion in between these two double stranded DNA. So now what happens then, indels, it causes the inactivation of particular gene. Okay, the next is the homology dependent recombination. See you can see here there are the two different double stranded DNA, and if we want to add a particular gene of interest here, so based on the homology of these area, based on the homologous recombination, this gene will be integrated into the gene of interest. So this is gene replacement. If we want to, you know, if we want to edit, if we just simply want to knock out a particular gene, then for us NHEJ is important. But if we want to add a particular gene of interest in the genome, then we have to go for the homology dependent recombination. So this is after creation of the double stranded break. So this is in general the protocol of CRISPR-Cas9. So what makes the CRISPR-Cas system the ideal genome engineering technology? As I've discussed earlier, the most important thing what is the basic thing here in case of genome editing technology is mainly the one protein which is going to identify the gene of interest that we are going to edit, and the next one is the endonucleases which is going to cut that gene. So the very this why the CRISPR-Cas has bring about the revolution in genome editing, because of its simplicity. If you have to target a particular gene, earlier in case of ZFN and Talon, you have to modify those protein which is going to identify the gene of interest. But here in case of the CRISPR-Cas9, you have to just simply design a guide RNA of 24 nucleotide, and that is very simple. Based on the gene of interest, you just simply there are various bioinformatics tools available, and you just place your sequence of the gene of interest, and they will suggest you the different guide RNA sequences. So that's why this technique has become very simple, and it could reach to the every lab and every individual crop species or the any living species where its application are possible. So this is the very important thing in case of the CRISPR-Cas system. So if you see the some of the advantage of the CRISPR-Cas high potency, that means the cleavage efficiency is very important. That means it could bring the cleavage, proper cleavage would be there of the DNA and its specificity. That means the off target that we were talking about, that it could target the some other portion in the genome. But here, in case of CRISPR-Cas system, it is uh, generally, in general, it is cleaving the in a particular portion that we are going to identify. This is a simple editing tool, as I've discussed. The main important point for the revolution, who has bring the revolution here. Okay, so there is ability to target the multiple DNA sites. So particularly, it is important from the polyploid crop. So in case of polyploid crop, for the same trait there are multiple alleles, right? And you for disrupting the particular gene, you have to disrupt the different allele, all the allele, different allele of that gene. So this is very important that we could able to target the all the alleles. So these are the different crops and where this CRISPR-Cas9 has been used. If we see the application in agriculture of this genome editing, it is can be used for the creating high degree of genetic variability at the precise locus. Okay, say for example, with the help of this, we can target even a single gene. SNP modification could also bring, so it will bring about the variability, genetic variability. So you can take a particular gene and you try to edit a different nucleotide every time, so it will bring about the different variation in terms of phenotype. So this is possible through the genome editing. This is potential tool for multiplexed reverse and forward genetic study. I've discussed earlier that what is forward and so there are different plethora of function could be done through the genome editing. Precise transgene integration at a specific loci. If we want to add, or if we want to integrate a particular transgene in a particular position as the genome, this is possible through the genome editing, particularly the CRISPR-Cas9, because the precision is important here. And developing biotic and abiotic resistant crop plants, we know that the how much these biotic and abiotic stresses, we know the climate change and its effects. So it is possible that with the help of genome editing, we could develop the biotic and abiotic resistant crop plants. It is a potential tool for the developing virus resistant crop variety. And you talk about the any trait like herbicide tolerance and anything. So this is possible through the genome editing, and particularly the polyploid crop like potato and wheat. Earlier it was this, when genome editing has been initiated, it could be used only in the diploid crops, but later on it could also be successfully applied in the polyploid crops like potato and the wheat. So this is it. And what are the way forward? Again, this is not the end of the genome editing. Again, I'll tell you, there are research is going on, and we know that there is a base editing and the prime editing. Today only we have submitted one manuscript in the regarding the prime editing. So the researchers has not stopped into the this CRISPR, though CRISPR-Cas9 is still utilized by the various researchers, but still there are a lot of research has been done in case of the genome editing, and now we have reached from genome editing, CRISPR-Cas based genome editing to prime editing. We have to take care about certain things like specificity, off targets. We have to be very cautious about the off targets. We have to develop a certain strategy or a kind of combination of the two system which are very specific. If we want to edit a particular gene and particular nucleotide SNP in particular position in the chromosome, that SNP only should be targeted. It should not be like it should go anywhere in the genome, and it should give any off targets. Polyploid crop, though these CRISPR-Cas has been successfully utilized, but is still in many crops like sugarcane and other forage crops which are polyploids, it is difficult to use this genome editing in these polyploid crops. So science is a continuous phenomena, and we have to continuous work for the refinement of this technology. Thank you so much. Thank you so much for your kind patience. If you have any question or query, you're welcome. Thank you. Viraj, are you there? Sriraj, are you there?
Yes, sir.
Okay, so I'm done with my presentation. If anybody is having any doubt or want to ask anything, they can ask. Or otherwise, you could share my number and my presentation, so that if they have any doubt, they can ask me in the future.
Yes, sir. Definitely, sir. If anyone is having problem or any doubt, then he can post his question in the chat box, and sir will clarify on it.
I think so. There are no questions. So
Those students are having doubts, please unmute and ask your doubts.
I understand that the topic which has been assigned to me, genome editing, it is very it seems like very complicated for the people, but I suggest if you just study and the basic things, it is not at all difficult for you to understand. The basic thing in genome editing is just the two things, two protein are involved. One is to identify the gene of interest we want to edit, and the second one is endonucleases, engineered endonucleases, which are going to cut the gene of interest. So this is the only thing here in case of genome editing. So is there any student from the MSc or PhD here?
No, sir. Most of the students are from first, second, and third year BC
Oh,
Agriculture.
okay, that's why I can understand. That's why I kept my presentation as simple as possible. I just wanted to give the overview. I didn't want to make it complicated. That's why I kept my presentation as simple as possible. Hope you people have understood at least what is genome editing and what are the different mechanisms and how it is working.
Yes, sir.
Okay, Sriraj, in that case, if there are no questions, we can move forward.
So shall we proceed to our next program, that is vote of thanks? So shall we proceed towards vote of
Yeah,
thanks?
sure,
The end of
yes.
session.
Please proceed.
Okay. Myself, Master Shriraj Kolte, take immense pleasure to deliver vote of thanks. First of all, I'd like to thank today's chief guest and speaker, Dr. Alok Shiv sir, for guiding our students regarding genome editing. Thank you so much, sir, for giving your valuable time and interacting with us. Secondly, I'd like to thank Honorable Dr. S.D. Masakar, sir, Associate Dean, College of Agriculture, Pune, respected Dr. A.B. Kamble, sir, Vice President, Students' Council, respected Dr. P.V. Patil, sir, Student Welfare Officer, respected Dr. Leena Shitore, ma'am, JRF SRF Cell in Charge, respected NSS Program Officers, Dr. Sabre, sir, Dr. Tamburi, sir, and Dr. Patil, sir, for providing such an excellent platform. And last but not the least, all the students present over here who gave the patient hearing. With the due permission of respected authorities and our chief guest, I declare that today's program is successfully over. Thank you so much, sir.
Thank you so much. And once again, I extend my